Hey everybody! Today we are going to spend some time talking about neurons. Specifically, we're going to talk about neuronal transport, and then we'll talk about classification of neurons. So in preparation for our next lecture on neurotransmitters, today we're going to spend a little bit of time looking at how neurons make neurotransmitter and then transmit it throughout that cell. Then we're going to move into a functional versus structural classification of neurons, and this will help as preparation for our upcoming lecture series on the peripheral nervous system. So let's get going. For today, you're going to need to have your pencils handy, possibly your erasers, because on worksheet 1.5, we're going to actually be drawing some of the different structural classifications or structural categories of neurons. And your science / college comics for today. The first one here, it says, if a neuron wore pants, would it wear them like this, along the axon there, like this, down on the axon terminals, or like this, up on the dendrites? Questions we should be asking ourselves. And then you might be too young to even remember this, but teachers in the past would make photocopies of worksheets, and each time they would grab it and photocopy it, each year would just get darker and darker. So you'd get these different iterations throughout time, and eventually it would just look something like this, where it would just be a dark circle for a cell, and they'd ask you to label it. And finally, inspirational meme from a student from last semester. Little pun on Canvas there. So, while we may never get to know how neurons wear pants, what we will know is how chemicals are transported throughout neurons with them a little bit. So, stick with me. So, you're all familiar now with basic neuron anatomy, but today we're going to focus on transport within that neuron. This is called neuronal transport. We're going to talk about two different kinds, fast axonal versus slow axonal transport. Then we'll get into that classification of neurons I mentioned. We'll talk about the 4 structural categories. And then we'll talk about the 3 functional categories real quickly, which we've already mentioned, and their locations within the body. And then we'll finish up with a little bit on synapses. We'll talk about chemical versus electrical synapses before we move on to neurotransmitters. Axonal transport, as you can imagine, is transporting materials along the axon of that neuron. This is really important for moving secretory vesicles within the cell. So we can move in 2 directions. We can move forwards or backwards. So forward direction, and when I say forward, I'm talking about moving toward the synaptic knob from the cell body. We call this anterograde transport. So that prefix antero meaning anterior, which is front or forward. So this is where we'll move newly synthesized material from the soma toward the synaptic knobs. Now we can also move in the reverse direction, retrograde transport, okay? And that's from the synaptic knobs toward the cell body. Retro meaning past or back. And here we're moving used materials from the axon for breakdown and recycling within the soma. Okay? Now I want you to think about for a second, what are some materials that may be moved toward the synaptic knob? And then what are some things that are going to be moved via retrograde transport back toward the cell body? So I'll pause for a second. You can come up with some answers and we'll revisit this. Okay, now that you've thought about it a bit, some materials that are going to move via anterograde transport are going to include things like enzymes, nutrients, and then what I alluded to when we were kicking off this lecture, neurotransmitters most importantly. Okay, some things that may be moved via retrograde transport. Oh, I kind of gave you the answer here. It's things that are going to be recycled, but those things are primarily going to be the vesicles. They're going to be broken down and recycled back within the soma so that they can be reused again to move things like neurotransmitters. All right, axonal transport can occur at 2 different speeds. We have fast axonal transport, which we'll walk through now, and then we'll compare that to slow axonal transport. Now as we go, I'm going to label different components so you're familiar with them. So first we're going to start looking right here, this small section of the axon. So that's what we're going to blow up, and that's what we're going to reference here. So we're just going to label this then the axon of the neuron. Okay. Now within that axon, we have a microtubule system. And you can think of this as kind of a rail-like system on which motor proteins are going to carry our cargo in either direction. So here's our first motor protein here, and this one is kinesin. Okay, so this is an anterograde motor protein. So this would carry maybe some neurotransmitter down to the synaptic terminals, so anterograde. Okay, and then down here, another motor protein carrying a vesicle there. This is dynein. And this is going to carry some materials in the retrograde direction. Maybe it's carrying a used vesicle in this case. Now, both of these motor proteins are going to use ATP to carry out their task. And once they're supplied, we can move that cargo. So we've dropped off here our neurotransmitter, and here we're going to drop off that used vesicle to be recycled. Okay. Now, some other things I should point out. While we say this is fast, this is all relative. So this occurs at about 400 millimeters per day. It's a little less than a foot and a half. And the reason that we use these microtubule systems and these motor proteins is because diffusion is just too unpredictable. Think about the longest neuron in your body going from the base of your spine out to the tip of your toe. It would take over 50 years via simple diffusion. But with these motor proteins here, we can get the products to
where they need to go within a reasonable amount of time.
Okay, a quick think pair share. What do tetanus, poliovirus, rabies virus, and herpes all have in common? Okay, this is perhaps a bit unfair because it's likely you may not know, but you do know it probably relates somewhat to axonal transport. Okay. So what they all have in common is they all enter at the synaptic knob and are carried to the soma by fast axonal retrograde transport. So here, let's take the polio virus, for example. So it's going to hitch a ride on that dynein motor protein and then be transported back to the soma where it's going to destroy the neuron. Okay, so polio, for example, that's that second picture here with the child sitting in the iron lung. This is destruction of motor neurons. So you'll lose your muscle control, you lose those respiratory muscles you need to inhale, exhale. And that's an example of one of these retrograde viruses here. Okay, now we can compare slow axonal transport to fast axonal transport that we just covered. Slow axonal transport occurs at about 0.1 to 3 millimeters per day. So this is really slow, especially if we compare it to the 400 millimeters a day of which fast axonal transport occurs. And so slow because it just results from the flow of axoplasm, or what we call as axoplasmic flow. Recall that axoplasm is just the cytoplasm of the axon. And another difference is that substances are only moved from the cell body or soma towards the synaptic knobs. Unlike fast, which we have both anterograde and retrograde, now we have only anterograde transport. And this is going to move things such as enzymes, cytoskeletal components the cell needs, as well as just new axoplasm itself. Now let's turn to the classification of neurons. So the first thing we'll do is visit the structural categories, and then we'll kind of apply some of that structural knowledge and revisit our functional categories. First, we'll start with structural classification. And this is as easy as counting the number of processes that are coming off of the soma, or the cell body. So for example, this one has 2 processes. Okay, let's practice this concept of counting these processes. So from left to right, how many processes are found on each neuron? Okay, so you can do this individually or work with the person that is sitting near you, and I'll give you a bit of time, but going from left to right, how many processes? Okay, well, let's take a look. So starting with that green neuron on the far left. Now remember, its processes include dendrites and the axons that are coming off. Those are all processes. So here we see that there are 1, 2, 3, 4, 5 dendrites, and then the axon. So that's 6 processes. If we move to the right now, we see there is 1 long dendrite and 1 axon. So that's 2. And here we have 1 short little process that leads to axon and dendrite. So that's 1. And finally, again, we have a 1 process. So we should have arrived at 6, 2, 1, 1. All right, let's move on. Now, to investigate the structural classification of neurons, we'll turn to your worksheet now, and you can follow along with me as we sketch the different classifications. So we'll start with multipolar, and then we'll move to bipolar, unipolar, and then anaxonic neurons. Let's begin with multipolar neurons. As that prefix suggests, we have many processes. In fact, these are what we think of when we picture a neuron in our head. 1 long axon and many dendrites. So, as you know, it looks something like this. We got a cell body, a long axon, axon terminal, the synaptic knobs, 1 axon, and then we have many dendrites coming out of that soma. So we can see here we have 1, 2, 3, 4, 5 processes, including these dendrites. So they're classified by many dendrites. And these include all motor neurons. All right, so that's multipolar neurons. Next, we have the bipolar neurons. And I'll switch up the color for us. Which again, you can probably guess there's going to be 2 processes with that prefix bi. Okay? So we got 1 cell body centrally located here, and there's going to be an axon coming off of it, 1 with your axon terminals. Okay? And then we got 1 dendrite. So 2 processes coming out of that cell body, 1 dendrite, and 1 axon. Examples of these include our special sense neurons. So these are going to be things like sight, taste, smell. Okay, all right, let's move on. Okay, up next we have our unipolar neuron. You probably guessed that has 1 process, 1, like a unicycle. Okay, and these are a little bit different. Here we have our cell body, and then we have a single short process. Oops, that should be an L. Okay. And then off of that, we have our axon that splits, and it splits in 2 directions. So we have, this is going to split into a central process. It's going to end in our axon terminal. Okay? And this often goes into the CNS, so you can remember it that way, the central process. And then on the other side of that cell body, we have what we call the peripheral process. And this goes out to the dendrites in your periphery. And this makes sense when we think about it because these are the neurons of your sensory neurons, like your tactile and touch neurons. So incoming sensory information here, again, it's not special senses, that's going to move along, and then it's going to enter your CNS. So it's going to go through the peripheral on the periphery, that axon, and then through the central process here. But this entire thing is your axon. It's just divided into two parts based on the location of this cell body. Okay? So we got single short process and one long axon. And as I said, the example of this is your sensory neurons. All right. All right. And finally, we have our anaxonic. So this prefix a meaning not. So these do not have axons. So you got your cell body, and then you got dendrites. Only dendrites coming off of these neurons. So the only processes are dendrites. And there is no axon. So where are these found? Well, you can probably take a guess.
These are our interneurons.
Oops.
And that's it.
And real quickly, since we're here, let's revisit functional classification. This will be a nice recap for you, see what you remember. So at functional classification, we classify these neurons according to the direction they propagate their action potentials. So again, remember that's toward the CNS or away from it, afferent, efferent. So the sensory neurons now, or those afferent neurons, they conduct that input from somatic and visceral receptors. So body, outside external world, as well as internal conditions, your viscera. And they send that to the CNS. And most of these, as we just learned, are unipolar. Motor neurons, or efferent neurons, they're going to conduct that output from the CNS to the somatic and visceral effectors. And all of these are multipolar. So that cell body originates in the CNS, and then from the CNS it goes to your somatic effectors, such as skeletal muscle. That brings us to interneurons. These are the neurons that receive and process information from the other neurons. So they communicate between sensory and motor. They're found entirely within the CNS. They make up the majority of our neurons, and they are multipolar, or as we just learned, anaxonic. A lot of those anaxonic neurons are found within your brain. Okay, now we can combine our structural and functional knowledge of neurons. So pull out your worksheet, and we'll start with sensory receptors. So we'll talk about our afferent neurons here. So we have a sensory neuron, and this takes sensory input, and this is going to pass through these unipolar, remember sensory tend to be unipolar, and that's where you got that central located cell body, which gives rise to ganglia. And here's a single ganglion, and that's going to move into the CNS where it synapses with an interneuron. And remember, interneurons can be multipolar or they can be the anaxonic. And I was actually just doing a little reading on anaxonic, and it's not that they don't have axons, they just think perhaps they're you can't discriminate an axon from a dendrite. So it looks like they don't have axons, although they do, they're just short. So it's kind of a misnomer. Anyhow, that synapses then with your efferent motor neuron. Okay? And This can give us motor output, such as controlling skeletal muscle. So that's just a quick recap of everything. Let's turn to chemical versus electrical synapses. Recall that a synapse is a place where a neuron connects to another neuron or an effector. It's the junction. And an effector here can be a muscle or a gland. So maybe a neuron synapses on a skeletal muscle or on an endocrine gland. Okay. Now there are two types of synapses. We have chemical and electrical, although chemical is far more common than electrical. So let's start with that one. First, let's go over the chemical synapse. I'll revisit a little terminology here to help clarify. So this is going to involve a presynaptic neuron. So I'll label this 1 here. This is before the synapse. The synapse is that cleft. And then we have a postsynaptic neuron. I'll label this as 2. And this is what that first neuron synapses to. So this is showing the dendrites then of another cell, another neuron. Okay, so presynaptic axon produces the signal, postsynaptic receives that signal, and the postsynaptic, this is generally a dendrite of a neuron. Okay, now the space between the two cells, we call that the synaptic cleft. It's a small fluid-filled gap. It's about 30 nanometers. To give you some reference, a human hair, the diameter is about 100,000 nanometers. So that is a tiny cleft. So with the chemical synapse and the synaptic communication that occurs, it involves a neurotransmitter diffusing across the cleft and binding the postsynaptic neuron. So you've learned by now the chemical electrical changes that occur to get that vesicle within the presynaptic terminal, that synaptic knob, to fuse to the wall and dump its contents. And then it has to diffuse across that cleft and bind to postsynaptic receptors on the postsynaptic neuron. And this does take a little bit of time. In fact, on average, it takes about 0.3 milliseconds. To put that into perspective, it takes you 300 milliseconds to blink your eye. So these events occur 1,000 times faster than you blinking your eye. With the electrical synapse now, we don't see that delay associated with the chemical synapse because we're not crossing that synaptic cleft. The presynaptic and postsynaptic neurons are physically bound together by gap junctions. So gap junctions they're made up of these 6 protein subunits that come together and form a physical tunnel that connects the two cells. It allows for direct communication. It's essentially like they're sharing their cytoplasm here. So there's really no delay. There's no neurotransmitter diffusing across a cleft. You get direct excitation between the cells. It's very fast. And it's just passing of electrical signal. It's found in limited areas of the brain and the eyes. We probably won't revisit it too much more, but you should be aware that an electrical synapse does exist. Right, the takeaway from today, we've seen that neurons use axonal transport, and they do this to move material within vesicles to and from that neuronal cell body. With anterograde transport, we're moving that synthesized material toward the synaptic knob, and with retrograde transport, this often involves moving used vesicles back to the body for recycling. We've also seen that neurons can be classified structurally based on the number of processes they have, and then functionally based on the direction the propagation of their signal progresses, away from CNS or toward CNS, or within. And then finally, chemical synapse is the most common type of synapse and involves neurotransmitter diffusing across that synaptic cleft, whereas the less common electrical synapse is more direct. It involves those connexons. Those cells are physically connected and occurs without delay. Okay, we'll see you next time.